My name's Katie and I'm a research technician here with the ex-situ conservation program at Kings Park Science. Ex-situ conservation is the preservation of plants outside of their natural habitat. The botanic gardens that you visit is a great example of ex-situ conservation, as the garden works much like a zoo to maintain a collection of diverse species from different regions across the state. Today you're going to see some of the science that supports the conservation work happening beyond the garden. In Kings Park Science, we have multiple groups of researchers working together. The teams I work with include our seed science team, who study the behaviour of seeds in and out of storage, the biotechnology team, who maintain the tissue culture and cryostorage collections, and the orchid conservation group that brings together all of these fields. My role is to support our research scientists in the technical aspects of their research, and today I'm working with the biotechnology team to harvest some shoot tips from Phylotheca basostyla in preparation for long-term cryostorage. Let's go! This is a day in the life of a Kings Park scientist. I always start my day by putting on my lab coat. The first thing that I'll do then is turn on my laminar flow. And this is a table with a constant flow of clean air, making sure that everything I'm handling is sterile and no dust or fungi spores or germs are falling into my sterile work. I give it a good spray with methylated spirits just to get rid of any of those things that might have landed during the night. And then I turn on my glass bead sterilizer. This is basically a safer alternative to the old school Bunsen burners. It operates at around 250 degrees to sterilize any tools I'm going to use. Then I will put on some gloves, again to try and stop bits of me falling onto my plant material as I'm working on them. Here I'm preparing the utensils that I'll need, including some scalpels and some forceps. Every person in the lab has their favourite forceps, and I have mine and I hide them here. We use surgical blades for extra sharpness, for a clean cut on our plant material, and I'll put the blades onto the scalpel handles and the tools will be ready to go. Then I'm going to grab a microscope. The shoot tips that I'll be harvesting today are quite hidden away, and a microscope will help me find them and cut them out without damaging them. This also gets a really good spray with methylated spirits, as I don't want to introduce any outside contaminants to our cryostorage collection. Then we're ready to grab our plant we are working with today from our tissue culture room. I'll be extracting shoot tips from 3 accessions. Accessions are genetically unique individuals in our tissue culture collection. The 4th accession will remain for another 3 weeks before processing. Moving material into cryostorage is a way of archiving our tissue culture collection. Tissue culture is resource expensive on both time and space in the lab, and all the jars you see here are subcultured regularly from 1 to 6 months, depending on species, to renew the nutrients in the jar. This maintenance is done by just me and our senior biotechnology researcher, and we have around 50 species to maintain. So cryostorage is a better long-term option for us. Plant material is stored at minus 196 degrees Celsius and in theory can stay alive for centuries. We only need to check them every now and then and for a couple of decades following. With all the prep work done, we're ready to begin. First step is to sterilize my tools long enough to kill off any germs, but not too long to burn my fingers. You'll see what I mean quite soon. Quite often the forceps are too hot to handle and I'll need to let them cool off before resuming my work. If it's too hot for me, then it's too hot for the plants. Using my forceps to grab out a plant, we can start harvesting our shoot tips. We store shoot tips in cryostorage because they are clean, healthy and full of living cells that can regrow into a whole new plant when they are raised from cryostorage in the future. Here I'm trying to remove all the leaves gently without damaging the shoot tip. The shoot tip is the very top part of the plant where new growth happens. I'm not cutting into the tip, rather than gently cutting the leaves off from around the tip before cutting the stem. We usually prepare around 200 shoot tips per unique individual for cryostorage. This ensures we have enough surviving material to allow for translocations and research purposes in the future. Today I'll be putting 50 shoot tips onto one agar plate, and then I'll wrap it in cling film to keep them moist and clean. Then I'll store them temporarily in a temperature-controlled growth chamber for 2 days in preparation for rapid freezing with liquid nitrogen. And this is how we prepare plants for cryostorage at Kings Park Science. Stay tuned for part 2 to see how we get those shoot tips into the dewar. Thanks for joining me in the lab today.